I must say that I'm really happy to be able to present in this this seminar series, and it's really an honor to me. So yes, I will be talking today about X-ray and neutron scattering of wood materials, and especially concentrating on the moisture interactions of the wood nanostructure. So I will first give a short introduction of myself and my background, and then I will talk about the wood structure and what we can learn from it with scattering methods, then more specifically about small angle scattering from wood, and then I will finish with some topics related to the role of water in the wood cell wall. So I did my PhD at the University of Helsinki in Finland, and during that time already I studied cellulosic materials with X-ray and neutron scattering. After that I went as a postdoc to Japan, to Kyoto University. There I investigated I was interested in the cellulose biosynthesis and studied that with bacterial systems. After 2.5 years there, I returned to Europe and I worked as a postdoc at Institut Laue-Langevin in France, and that's where I used a lot of small angle neutron scattering and also developed a model to interpret the small angle scattering data from wood. And in 2018, I returned to Aalto University, or to return to Finland, and started a postdoc at Aalto University, and mostly working on the moisture behavior of wood, and again with X-ray and neutron scattering. So, and currently I'm a academy research fellow at Aalto University. A few words about Aalto University. It's a relatively new university, started in 2010, but it was formed by a combination of a former technical university and an art school and a business school. So it's like a multidisciplinary science and art community that combines these different fields. And it's located in Espoo, which is right next to Helsinki. So this is the city center of Helsinki. And nowadays you can take a metro from there to get to Otaniemi, where the Aalto University is located. There are also a lot of companies and for instance the VTT Technical Research Center of Finland in the Otaniemi campus. There are like 12,000 students and 4,000 staff members, and quite a large share of international faculty staff. And like I said, I am now working as an academy research fellow, so that's a 5-year position funded by the Academy of Finland. And with this position, which started just last year, I have been able to start my own research group, which has the name Bio-Based Material Structure. And it's part of the Department of Bioproducts and Biosystems in the School of Chemical Engineering. We are currently 3 members in the group, so it's myself and Aleksi, a third-year PhD student, and Enriqueta, who just started her PhD studies this spring. And in the group, we are concentrating on studying concentrating on the structural characterization of bio-based materials, especially with X-ray and neutron scattering. And we also do some development of the data analysis methods. And recently, or in the current projects, we are mostly interested in the nanostructure of wood and how it behaves with moisture. And these are the topics that I will be telling today also. And but first we should look a bit in the structure of wood. So you probably all know that wood is a highly hierarchical natural material, and it's formed of, or this composite structure in the cell walls is formed from cellulose molecules, which are aggregated into crystals or like semi-crystalline microfibers. And these microfibers are then bundled together with hemicelluloses and some lignin, and then they are in the secondary cell wall of the wood cells or wood fibers. And the properties of wood as a material are closely related to the nanoscale structure. So that includes, for instance, the mechanical properties of wood and its moisture behavior. So it's quite important to understand the structure at this level. At the same time, many of the new applications of wood and plant-based materials in general, they are based on the nanoscale building blocks of the wood cell walls, so the cellulose microfibers, and especially based on their interactions with water. And that's why it's crucial to remember that water is also one component of the cell wall nanostructure. Then there are many methods that can be used to characterize the cell wall nanostructure, and I'm mostly working with scattering methods, and they have some certain benefits over other methods. So in one experiment you can obtain a quite a nice average structure of a large ensemble of nanostructures. So you measure just a macroscopic sample volume and you get statistically significant data on the structure. It's also possible to cover multiple levels of structural hierarchy with these methods. So you can go from the molecular level to the level of microfibers and even microfibril bundles. And maybe one of the main benefits of the methods is also that they are really flexible regarding the form of the samples. So you can measure a solid piece of wood or a wet piece of wood or a gel or whatever, almost whatever. And you can also vary the sample environment, so you can change the humidity, like in this setup, for instance. You can change the humidity or temperature or all kinds of parameters. And something that is also possible are the time resolved studies, and that's especially with the synchrotron sources. You can get to time resolutions of below 1 second. So you can study quite fast processes. Then a few words about the principles of the scattering methods. So they are based on the interaction of X-rays or neutrons with the sample. And in the sample, these X-rays interact with the electrons and neutrons interact with the atomic nuclei. And basically these X-rays and neutrons in scattering, they are sensitive to the spatial variation of the scattering length density. And the scattering length density is, for X-rays, it's simply equal to the electron density, and for neutrons, it's slightly more complicated. But anyway, so if we have here a scatterer, it can be a particle or just even an atom. We have X-ray or neutron radiation coming towards it with the wave vector k, and then this wave is scattered at different positions inside of the sample or inside of this scatterer or the particle, and then it's these spherical waves that interfere with each other, and then the sum up wave is the one that we detect on the detector. So depending on the locations of the scattering points here inside of the particle, we get a certain type of interference pattern on the detector. And this tells us about the structure inside of the sample. And we often present the data as a function of either 2 theta, this angle here, or the scattering vector q, which is derived from these wave vectors shown here. But more on a practical side, so if we have a wood sample like this and we show an X-ray beam, or can be also a neutron beam to it, at the close distance from the sample, we can observe the scattering at the larger scattering angles. So that's called the wide angle X-ray scattering. And then if we move the detector further, or we possibly have a hole in the detector in the middle, we can detect the scattering at the smaller angles. So this is just a geometrical reason. And these two small angle and wide angle scattering patterns, they contain information on different structural levels in wood. So specifically, the wide angle scattering originates from the crystal structure of the cellulose, or in the cellulose microfibers. We can see there the different diffraction peaks corresponding to different directions in the crystal. Also, water produces a contribution here. Then at the smaller angles, we see scattering from larger structures, and this means the structure of the cellulose microfiber and how they are packed together. So from the wax data, we can obtain the lattice spacings, so distances between the molecules in the crystalline lattice, and the size of the crystal. And then from SAX data, we can obtain the diameter of the microfibers and the packing distance between the microfibers. Also some other information, and that's what we will discuss next. So in the small angle scattering from wood, the different hierarchical levels contribute to different regimes of the scattering vector Q. So this is now a small angle neutron scattering pattern of wood, so it's highly anisotropic, and we are now looking at the scattering here at the equator. So at the larger Q values, we have the scattering from the microfibers, which produces this kind of shoulder feature, or some kind of bump in the curve. And then at the smaller Q values, we see scattering from larger structures, which can be some kind of pores or the hollow spaces of the wood cells, inside the wood cells. And as you can see here, also the SAX and SANS data, they look slightly different, and that's because of the different principle of the scattering. So that the X-rays are scattered by neutrons, and X-rays are scattered by electrons, and neutrons are scattered by the atomic nuclei. So they basically see a slightly different landscape of the scattering length density. And one nice thing with the neutron scattering is the possibility of contrast variation. So if we have a sample with wet wood in 100% H2O, we have in the cell wall, we have the cellulose microfibers, which are surrounded by a polymer matrix, which includes the hemicelluloses and lignin, and also water. And then there are some pores or voids filled with water. And these are the cell lumina. Then if we dry this sample, what happens is that the water is removed from here, and then there's basically no contrast anymore between the cellulose microfibers and the cell wall, or the microfibers and the matrix, so we cannot distinguish any structure inside of the cell wall. But we see a strong scattering from the interface between the cell wall and the air-filled pores. And with neutron scattering, with this contrast variation, we can actually choose the ratio of H2O and D2O and use that to increase the contrast between the cellulose microfibers and the matrix. So that's why we typically measure the suns in 100% D2O to see the best possible contrast. It's also possible to use a condition called contrast match. So that's a condition where the matrix has the same scattering length density as the cellulose microfibers. So in that case, we lose the contrast almost completely. And to analyze the small angle scattering data from wood, I developed this model called WoodSAS, and it's used to interpret the equatorial scattering data, suns or SACs, from wood samples. And here in the equator, we see the scattering from the microfibril cross-section and their packing. And so this model has 3 components, and I can say that it's a combination of previous works. It's not only invented by myself, but it's tailored. I kind of tailored it for wood samples. And here the cellulose microfibers are assumed as or approximated as hexagonally packed cylinders based on this paper, and they have a diameter of 2r and a packing distance of a, and both of these have a polydispersity. Then the second term here is slightly more vague, so it's not entirely clear what it corresponds to in SACs data, but I will show in the following that in the SUNS data we have been able to link it to the diameter of the microfibril bundles. And then the third term here, that's this power law term here, and that originates from the surfaces or the interfaces between the cell wall and the cell lumina. And like I said, this works for SUNS data as well as SACs data. And now, what kind of information can we get with this model? So we can determine the microfibril diameter in wood, and the results show that in softwood it's about 2.5 nanometers based on SACs, and that's similar result as has been determined with slightly different methods here. For instance, like checking the minimum of this form factor here. And then in SUNS, the diameter of the microfibers is slightly smaller, especially in deuterated water, and that's probably because the surface hydroxyl groups become deuterated when they are in contact with deuterated water. And these values agree quite well with the 18-chain model of the cellulose crystal. Regarding the microfibril packing, it looks like in softwoods, in wet state, the microfibril packing distance or distance between the microfibril center points is about 4 nanometers, and in hardwoods it looks to be a bit smaller. And when you dry the sample, the distance between the microfibers decreases, especially in the softwood. And this is something that we showed also with time resolved neutron scattering, so it has been shown also in other studies, but I think this is the first time-resolved SANS experiment of a drying wood sample. So we measured at the ILL, the 11 beam line, and we had a sample of wood that was first in deuterated water, and then we took it out and just let it dry in the experimental hall, and so in ambient air. And from the data, we could see that the distance between the microfibers remained the same for quite a long time, and then at some point it started to decrease. So we interpreted the data so that the free water is evaporating first, and then once the water inside of the cell walls starts to escape, then the distance between the microfibers decreases. And this was also linked to the diffusion coefficient of water in the cell wall. Okay. Then I mentioned about the second term of the Wood-SAS model, and that's the one that has been linked to the microfibril bundles. And this study was done by using polyethylene glycol. And firstly, if we have a native wood in deuterated water, and this is the term of the microfibers, and this is the term of the power law, so from the pores, and if we subtract them from the experimental data, then what remains is this kind of quite a smooth contribution. And so this contribution was then fitted with the Gaussian function in the Wood-SAS model. And it's possible to use the Gaussian or interpret the Gaussian function as a linear law, which is a well-known method for determining the size of the scattering particles. And in the case of native wood, it would correspond to some cylindrical structures with the width of about 18 nanometers. So that's in the same scale as the microfibril bundles. And like I said, we used the polyethylene glycol, so we impregnated the cell walls with this PEG. We had 3 different molecular weights of that, and what we saw then in the scattering was that because the contrast from the cell lumina was decreased, so this term decreased, and then this one was shown more clearly, and at the same time the peak from the microfibers became sharper and shifted to larger Q values. So we interpreted this data so that the PEG is it penetrates really to the cell wall outside of the microfibril bundles, but not inside of them. So then and it's very hygroscopic, so it probably kind of sucked out the water from the microfibril bundles and made them shrink, so that the distance between the microfibers decreased and also the overall diameter of the bundle decreased. So with this interpretation, we can use this term to determine the diameter of the microfibril bundles in basically intact cell walls. So we don't need to do anything to the cell walls. We can just put the sample in the beam and well, we have to use deuterated water, but that's anyway just water. So by doing this fitting, we can calculate the diameter of the microfibril bundles under certain assumptions. And this seems to vary. For wet softwoods, it's a bit larger than hardwoods, and especially it's smaller in dry softwoods. But this is quite difficult to determine because the contrast is very low in the dry state. But anyway, these values agree quite well with values found in the literature. Okay, then the last topic is now the role of water in the cell wall. And as we all know, water is really important for the material properties of wood. So wood is very sensitive to moisture changes. It swells or it can shrink and produce cracks. And often the processing of wood takes place in an aqueous environment. So it's important to know where the water is located in the nanoscale structure of the cell walls. And water is also really important in, for instance, self-assembly of cellulosic nanomaterials. And to study this, the location of water in the cell wall, we conducted some neutron scattering experiments again at ILL. And normally we would measure the samples at fully deuterated water, and that's the case where we have the maximum contrast between the crystalline cellulose and the water or the matrix. But in this case, we had measured the samples first at the contrast match condition and then transferred or exchanged the water to 100% deuterated water and then started the time resolved experiment. And in that we can see that these features are slowly emerging from the, or even not that slowly, actually quite quickly emerging from the background. So like this. So that means that we increase the contrast inside of the cell wall when the water is exchanged there at the level of the microfibers. And we took the equatorial anisotropic scattering intensity and fitted it with the Woods model, and especially only this cylinder term, because we didn't have the data at lower Q values. And we can see here that the structural parameters, the diameter of the microfibers and their packing distance, they remain about the same. But what changes is this scaling factor A here, which is proportional to the square of the electron density or the scattering length density difference or the contrast. And we can see that this value increases and it reaches a plateau after about half an hour. So this means that the water was exchanged inside of the cell wall in this piece of wood or these pieces of wood within about half an hour. Also in the same data we can see scattering from slightly different length scales. So we can see scattering from the level of the microfibers and the microfibril bundles. And the question what we wanted to solve was that if there's a difference in the exchange of water at these different structural levels. And so we looked at analyzed the azimuthal profiles of these curves at different Q ranges, and then the data is shown here. So we can, and for comparison, we also used the scaling factor from the Woods model. And in all of these, we could see that the water is exchanged within about half an hour. So, and even by fitting a function here, we didn't see much difference here. Only if you really look at the values like 5.9 and 6.1, this value is slightly larger, so that it could indicate that the water at the level of microfibril bundles is exchanged faster than inside, but this is not really a clear result. Then I have one last topic. about the moisture effects on cellulose microfibers, and this is a very recent work. So it's known in the literature for a long time already that the diffraction peaks of cellulose shift when you change the moisture. For instance, if you dry the sample, and sometimes they have been reported to broaden also with drying. And there have been several mechanisms explained or trying to explain this, and for instance for the 200 peak there are these two at least these two possible explanations. So the first one is that when the matrix polymers swell, or the matrix swells with the water, it kind of presses the microfibers in the lateral direction, so the distance between the 200 planes decreases with moisture. The other a bit kind of opposite explanation is that when the matrix shrinks while the sample dries, then it somehow pulls apart the 200 planes, and the effect is the same, but it's a kind of opposite mechanism. And to study that, we did these experiments with the new Xenox SaxWax device at Aalto University using a humidity chamber, and we made a moisture ramp from a fully wet sample down to relative humidity 10% and then up to 95. And we could see quite consistent changes, especially when we converted these relative humidities to the or we measured with dynamic vapor absorption the moisture content of the samples. So we could see really reversible trends in the lattice spacings with the moisture content. And also the crystal lid size was larger, so the diffraction peaks were narrower at higher moisture contents. To study this then in more detail, we have had already for a couple of years this project that is done in collaboration with BTT, and it's especially Dr. Antti Paajanen, who is a modeler, and it's funded by the FinCERES flagship of the Academy of Finland. So there we use molecular dynamic simulations of the microfibril bundles, and then we can also compute the scattering data from these models, and that has many purposes. So we can refine the molecular models according to the experiments, we can get new insights into the experimental scattering data, and we can test hypotheses. So this is an example of a microfibril bundle. It's a periodic model, so this fibril continues here, and so you can see it swelling here. These are the hemicelluloses, and these models they produce relatively nicely the packing distance of the microfibers. There's quite some spread in the literature values depending a bit on the method, if it's SAX or SANS, or also the model used. But anyway, this swelling behavior was reproduced well. And then we have the experimental wax data. And now the nice thing with the models is that we can compute the scattering data from the model structures. And this is the data resulting from the model that you just saw. And so we can observe the same shifts of the peaks here, and they fit quite well to the experimental data. Also, it's possible to determine the distances of the molecules from the models themselves. So that's this solid line here. Also, we analyzed the broadening of the diffraction peaks, and they produced similar trends as in the experiments. So what then actually explains this shift of the peak and the change of the um, peak broadening is that that actually when the microfibrils or when the microfibril bundles dry, the microfibrils get closer to each other, and then it looks like somehow the surface chains of the microfibrils are starting to interact with the neighboring fibrils and also somehow with the hemicelluloses. So this kind of destroys the ordering of the crystals here at low moisture contents. And that's also clear from if we look at the D-spacings determined from the model and look at their distribution as well. So for instance, in this 200 direction, the lattice spacing fluctuates really a lot at low moisture contents. So what I want to say with this is that the crystals seem to really deform at low moisture contents, and this fibril-fibril interaction is really important cause for these crystal-like deformations. So to conclude, I hope I could show that the X-ray and neutron scattering are really powerful tools for structural studies of wood and also other cellulosic materials. They can provide a representative average structure over multiple hierarchical levels, and it's possible to utilize varying sample environments like different moisture contents and also to do time-resolved studies. And still, I can say that the developing of the accurate interpretation of the scattering data is an ongoing effort. And as the last thing, the water is really an active component in the cell wall structure. And then with this, I would like to thank all the people who contributed to this work or these works, and thank also to the funding sources. And thank you for your attention.
So now we open the floor for questions.
Do you want me to start?
Yes, start.
Thank you for a very nice presentation, Paavo. It was fantastic to listen to it, and it gave us a really nice overview of the subject. I was thinking about the diffusion. If you go back to slide 16, 17, something like that.
Yes. Here.
I think it was 16 or 17 in the beginning. I noted that 16. Here.
This, yes.
Yeah. Then you talked about that you had in the figure there, the sun's figure, you dried it, and the moisture was diffusing out. When you measure, how difficult? I mean, you measure an average, and the diffusing is over many length scales there. So how is that influencing the dimensions of the wood piece you are measuring? Can you tune it so a bigger wood piece will make this speed slower?
Yeah, I think yes, of course the size of the wood piece makes a difference as well as many other factors. So this experiment was not done under any controlled circumstances. It was more like just to show that it's possible. And maybe if we would do it again, we would do it in a more controlled way and really like controlling so that the evaporation happens only maybe on certain sides of the sample, and maybe there could be some kind of flow of air or at least a control of the flow of air and factors like that. So that was maybe the challenge here to actually also to interpret the data, because we were not fully sure that the water is evaporating evenly from all parts of the sample, and probably it didn't.
Thank you a lot. We have some questions in the Q&A and chat. Alexander, do you want to read them?
And we also have a raised hand from Paulina.
Oh yeah.
First,
okay.
We'll allow you to talk.
Hello, thank you very much. Very nice presentation. I mostly had a question about contrast of cellulose and D2O. What kind of SLD value did you use?
You mean for the
For the small angle neutron scattering experiments?
For the contrast match condition? Yes. 35% D2O and 65% H2O.
And then the rest of the experiments you performed in 100% D2O, right?
Yeah, or well, it's not really 100% D2O, because we had the sample first in this condition, and then we just exchanged the water here. So some of the H2O remained inside of the sample, but I think it was still maybe 90% D2O. Okay.
And what SLD value did you assume was in sort of 100% of D2O? Like, did you assume that all of the hydrogens were deuterated, or?
We didn't really calculate the value, so it was not needed for this. But there's a certain fraction of the OH groups that become deuterated.
Okay, thank you.
So thank you. So from the chat, we have a question from Lengwan Li. So says, nice work. Did you try deuterated PEG? What are you expecting to get in your scattering curves if you would use deuterated PEG?
We didn't try deuterated PEG in this case. Of course, yeah, it changes the contrast, and in this graph it would change it to the higher scattering length density. But it's quite a difficult thing to say how the data would look like without actually measuring it.
Okay, and then we have from Lars Wågberg. So thank you much for your excellent presentation. Regarding your last example with the mixing of the chains from different fibrids at very low moisture content, how low is low? Is this reversible? And he actually assumed that this was the lignified fibers.
Yeah, well, this was native wood, so lignin was there, and the low moisture content, now I don't remember the exact value, it's probably around 5 or even a bit less. So it was, the sample was at relative humidity 10%. So I think it could be, with some setups, it could be possible to go even below that, and it would be really interesting to see, and also to see if there are some irreversible changes taking place there.
And then we have from Lennart Salmen. Paavo, can you explain why moisture increases in the RH region, why the moisture increase in the RH region not only compress the cellulose crystal, but also strains the crystal to a larger extent than would be expected from the Poisson ratio?
I have to think a bit on the question. So, not only compress the cellulose. Yes, so if it refers to the longitudinal distance here, that's something that our models, they reproduce this behavior, but we don't know exactly why. So it's one challenge there is that the models are of limited length in the direction of the fibril, and yeah, it's a tricky question.
Okay. And then a final question here from Ebru Sarioglu. If you do this experiment for the lateral surface of wood, not cross-section, what can we see?
Well, in this case, I do not fully understand if it's related to this. Here the sample is really like a stick of wood, like a match, similar size and shape. So it is we don't have a cross section in that sense here. It's just that in the drawings I have presented them like this, but that's also because the data that we get is mostly on the equator of the scattering pattern, so that it corresponds to the lateral cross section of the cell wall.
Okay, and then a question from Linnea Björn. In your work, most analysis from the wood SAS model is based on the equatorial streak in the scattering pattern. In most materials produced in FibRe, the fibers are taken out from the wood structure and altered, and most often arranged in a random fashion in the final material, giving rise to symmetric scattering. Can the wood SAS model still be used even if there is no global alignment on the fibers or microfibrils?
That's a good question. I have not tried this model properly on any processed wood material, so that's something to be done. But according to my previous experience, it looks like the scattering from pulps and these kind of processed fibers is somehow, it has weaker features than the data from wood. So if I show here that we don't get this kind of clear peak or not even a clear shoulder like this. So the structure in wood is somehow more regular than in the pulp fibers, for instance. But it's something that should be done in the future.
We also have a question from Artem. Very nice presentation, thank you. What is the spatial accuracy of the diameter and interfibril distance of the measurements? And you have a second one, but how have you looked at the changes in the scattering during the deformation, in particular beyond the yield limit?
Well, to the latter question first, we have not studied any deformations, so that's also something to be done in the future. And for the spatial accuracy of the diameter and interfibril distance, there are different sources of error and different margins of error that you can think of. So one is from the actual fitting of the model. That's usually very small error, but then there's other error, for instance, from slightly from how you process the data, or if you had to do some kind of rebinning or the quality of the data. That's one thing. And then of course the natural variation of these parameters. At least if you measure from the same tree and same parts of the 3, then there's very little variation. But it's like 0.1 nanometer or less variation. But otherwise there can be more variation.
Okay.
And that's something that should be studied as well. That what is the natural variation and how do these features vary in a tree, for instance, in different parts of a single tree.
Do we have more questions in the chat, Alexander? Or was this the last one?
I think we have covered all of them.
Fantastic. Can I go back to Linnea's question about the fibers? So do you have an idea how to use your model on fibers and how one could, is it possible to, do you think the problem with fibers is that they're not aligned, or do you think it is the restriction that would more create them more restricted orientation?
Well, at least I would say that the orientation is something that you can change just by manipulating the sample or preparing it in a certain way. So then it's more about the contrast and the regularity of the structure. Because as far as I understand, if you do pulping to the cell wall, then there are, for instance, some pores that are formed by the removal of lignin, and they contribute to the scattering as well. So there you have then a system of where you have also air-filled pores inside of the cell wall, and they have roughly similar size as the microfibers and microfibril bundles. And then also there can be some aggregation of the microfibers, which might lead to a more variation in the diameter of the fibrillar units.
It was fantastic, nice presentation, and also very funny questions. So we're very, very happy with that. Alexander, do you have any smart concluding words?
No, I think it's super to have had you here virtually. I hope we can also host you live in the Treesearch environment, both at KTH and Chalmers, and also at when we are up and running with the Formax Beamline. I really hope that you will be one of the principal users in the future. And I think this also shows the opportunities later for using the European spallation source, where we get considerably more neutrons, which shortens the experimental time. So we can do much more systematic studies or go into more complex environments, enhancing data quality and so on, but also maybe thinking in completely new ways of doing experiments. So I really look forward to a continued interaction from the FibRe and Treesearch environments with you on these topics. So thank you very much for participating.
Yeah.
Thank you also very much for inviting me, and I'm looking forward to use the scattering facilities also that you will build in Lund.
Yeah. Thank you. Bye bye.
And should we say just that this was the last webinar before the summer?
Yeah.
Or if it's, or maybe it's already summer, but with this series we'll continue after summer, and there will be more information. That's what you promised, Annette, at least.
Absolutely.
We'll continue. So thank you, Paavo, for the presentation today. Thank you for you who have been listening here on Zoom and discussing, and who have been following on YouTube. So have a nice summer, and welcome back later in the autumn.
Thank you.
Thank you. Thank you both, Paavo and Josephine. Thank you a lot.
Thank you.
Bye bye.
Bye.